I'm going to explain how geneticists use genetic techniques to identify alleles, loci, that might contain incompatible alleles. This concept is sometimes known as transmission ratio distortion and segregation distortion. Understanding how this analytical process works requires us to review a little bit about meiosis and the generation nomenclature in genetics. So the way that this diagram is organized, and I'm going to be filling this in as we go through this demonstration, is I've got generations listed on the left with the P0 or parental generation shown at the top, and then I'll diagram individuals from an F1, the offspring, and then the F2 generation, the grandchildren. And the somatic genotypes, the soma, are cells in the body that are any cells other than the gametes, which includes sperm and eggs. And I'm not indicating here which type of gamete is a sperm or an egg. It doesn't really matter for this example. So what I'll diagram here are genotypes, and I'll diagram cartoons of chromosomes from two different individuals of the same species, parent A and parent B. And inside each nucleus or cell of each somatic cell example in each gamete, I'll draw cartoons of two different chromosomes, which I'll call chromosome 1 and chromosome 2. So here in the PW generation, I'm assuming that parent A has particular genotypes of chromosomes that are diagrammed as being red chromosomes. And in this case, red stands for a particular DNA sequence that's different from the chromosome sequences found in the other individual of the same species, parent B. So while these are meant to be two individuals, A and B, from the same species, the different colors of their chromosomes indicates that those chromosomes can be distinguished based on the DNA sequence of those chromosomes. In other words, the parents are genetically different. And so I'm going to use the colors of the chromosomes to track what happens when these chromosomes undergo meiosis, the first step, the arrowheads, and produce the gametes from individual A and the gametes from individual B. And then we'll see what happens to those chromosomes when those two gametes fuse at fertilization to produce F1 offspring. And before I go any further, I should mention that it's an extreme assumption, which is almost never going to be true, that any parents would be homozygous. That is, both copies of the same chromosome, shown as the two horizontal lines, two copies of chromosome 1, the shorter chromosome, and in this case, two copies of chromosome 2, the longer, would be genetically identical, which I've diagrammed here as both of them being the same color, both being red. That's very, very rare, probably impossible in most species, but we use it here as an assumption to make the diagramming easier. So here what we can see is, in meiosis, what happens is the gametes have half as many chromosomes. They have one of each instead of two of each as in a typical somatic cell. So in the process of meiosis, one of the two copies of a chromosome will be put into the egg or sperm, the gamete, and that's true of every single chromosome. So in this case, that's how those products of meiosis, the gametes, are produced. And so because individual A only has completely red chromosomes, there's no way that any gamete other than a totally red gamete, sperm or egg, can be produced. And the same is true of parent B. So if we know that parent A is, as I mentioned earlier, totally homozygous, all of their chromosomes are identical. Chromosome 1, the two copies of the chromosome are exactly the same. The two copies of chromosome 2 are exactly the same. Then the geneticist can predict, we know, that there's only one possible type of gamete. It's going to have the same genetic makeup as the parent somatic cell. And that's why I note here that the gametes that I've drawn in the P0 generation are all of the possible haplotypes. Haplotype being a fancy word for genotype of a haploid cell or gamete. So in this case, we know that's all of the gametes that parent A will make are going to be like this. Red chromosome 1, red chromosome 2. And parent B can only make blue chromosome 1, blue chromosome 2 type gametes. Likewise, you should be able to predict what's going to happen when these two gametes fuse to reproduce a diploid cell through fertilization. So what is going to be drawn in the somatic cells of the F1 offspring when these two gametes fuse at fertilization? The offspring inherits a red copy and a blue copy of chromosome 1, and a red copy and a blue copy of chromosome 2. So in this case, in this specific type of cross, each F1 offspring, here I've shown two different individuals, they're genetically identical. And that's because all of the gametes of the P0 parents in this case are identical. So every single fertilization event between parent A and parent B will involve red chromosome 1 and red chromosome 2, and blue chromosome 1 and blue chromosome 2. So all of their F1 offspring will be genetically identical to each other. And here is one of the first critical concepts of understanding how to use genetic crosses to look for genetic incompatibilities. In the F1 generation, if we turn our attention to the graph on the right, what a geneticist would do to understand what the expectation is for alleles, the types of chromosomes that are present in offspring, which is what we're going to eventually do experimentally in the F2 generation. Here in the F1 generation, we can look at each spot on each chromosome. So we could look at, for example, the left end of all of the chromosome 1s that exist in this generation, in this case 2 individuals. And we could plot at that locus on chromosome 1, the first little segment, what percentage of those 4 chromosomes are red. So we have 1, 2, 3, 4 chromosomes, 4 copies of chromosome 1 in this generation. Half of them are red. Right? So we've got A and B, for example, little a, little b, just using a different method of counting chromosomes. So we've got 2 copies of a red block of chromosome, and C and D, 2 blue. So 50% of the 4 chromosomes are red. So we can plot that in the graph on the right. The top would be 100% red, the bottom is 0% red, so we're at 50% red. That first little segment of chromosome 1 is 50% red. And then we can move to the next block, and it's still 50% red, and we move that way across the small chromosome, chromosome 1, and every time we look, when we count up the red and blue across all of the copies of the chromosomes in that generation, we find that there's 100% of the time, rather, it's 50% red, 50% blue. Again, this shouldn't be a surprise, because we know that the contributions of genetic material from the gametes into this generation were 50% red and 50% blue. So this first step is pretty trivial, but it's important to note because it sets our expectation. And evolutionary geneticists use this as the null hypothesis, or expectation, that if you start with a hybrid individual, or a series, a population of hybrid individuals, that have a 50% red genotype, then if nothing evolutionarily happens is selected for or against in terms of the genotypes of these individuals, then as time passes, in an ideal situation, like no genetic drift, in the F2 generation, we should still see, of all of the individuals in that population in the F2 generation, however many there are, we should expect to see the same frequencies. On average, half of the chromosomes should be red and half blue. And that's why down here in these graphs that I've created, when we look at the F2 generation, there are these gray lines. Those are these expectations that we came up with from the F1 generation. That all else being equal, if no genetics are being selected for or against, no genotypes, then the allele frequencies, the percent red that every part of every chromosome should stay the same, 50% red, 50% blue. In other words, if we were to see a bias or some sort of plot that showed that there were deviations from that expectation, those deviations, segregation distortion or transmission ratio distortion, is suggestive that there could be selection acting on certain genotypes. This would mean, for example, if we saw a peak there where 100% of the organisms in a population in one generation only had red alleles, that might tell us that it's beneficial for those organisms to have red alleles, or similarly, it could tell us that it's bad to have the blue version of that chromosome, and that's why there are no individuals left in the population or existing in the population that have the blue allele. So in summary, this is the concept of transmission ratio distortion. We generate an expectation, and then we experimentally look at hybrid genotypes. We genotype a bunch of individuals, and then we plot the distribution of genotypes across one or multiple chromosomes, and we look for regions where there's statistically significant divergence from the observed values and the expected values. So now it's time to think about the second round of meiosis and what happens when the F2 generation is produced. And before we get there, there's one other concept to know about or to remember. And that's the fact that when meiosis occurs, chromosomes can recombine with each other. And simply, what that means is during the process of meiosis, chromosomes can break, which I'll diagram with a little X, and exchange arms, pieces of chromosomes. So in this example, during meiosis, what would happen is that pure red chromosome in the F1 individual would break into two parts, so the left half of the chromosome and the right half of the chromosome, and they would physically recombine or stick together with the equivalent pieces of the blue chromosome that were created when both copies of the chromosome broke. So this is something we didn't see in the P0 meiosis, creating the F1, because recombination still happens up here, but we can't tell because what does a blue chromosome look like when it recombines with another blue chromosome? You produce yet another all blue chromosome. So meiosis was seeing recombination, but we can't detect that genetically when a parent is homozygous, when their two chromosomes are identical. Now in the F1 generation, we have a heterozygote, an individual whose two copies of the same chromosome, chromosome 2 for example, are different. We can detect when recombination happens by seeing chromosomes that have some of the red DNA sequence, and then the rest of the chromosome carries on with the blue version of the chromosome sequence. So now we can see how I've diagrammed possible, really important, example recombinant haplotypes. Recombination is a very complicated process that can create a lot of different genetically diverse chromosomes. In this case, we could imagine that recombination in chromosome 1 happened there, on chromosome 2 happened around there, chromosome 3, chromosome 4. And I'm leaving out a lot of subtle details about how recombination happens and about how meiosis works. So for more information, check out videos or other resources that explain those concepts in greater detail. But the point here is that we have example gametes of the F1 individuals. So one F1 sperm, for example, could be a little bit more explicit. Let's say that's a flagellated sperm, and let's say that's an egg, singular, an ovum. Then the sperm has a recombinant copy of chromosome 1 and a recombinant copy of chromosome 2, and the same is true of the two versions of the chromosomes that are in the ovum from parent B. So now we're talking about the F1 individual. So let's instead refer to them as, say, individual C and individual D. So individual C produced many, many sperm, certainly in their lifetime, but here's just one example of a possible haplotype, genotype of the sperm. And again, individual D, a female, produces many ova in her lifetime, but this is one example. And like before, we know, based on how I've drawn these gametes, what the F2, their offspring of C and D, the F2 child, grandchild of the P0 generation as well, will look like. It'll be the 2 copies of chromosome 1 and the 2 copies of chromosome 2. So right now, we're at the point where we're starting to look at F2 genotypes. And remember, the idea here is to look at an F1 to generate the predicted or expected frequency of chromosome fragments, of alleles, red versus blue. And what we're going to want to do is to look not just at one F2 individual, but lots of F2 individuals, and then plot the average percent red across, say, chromosome 1 and chromosome 2, and look for deviations from this gray 50% expected line. So before we get to multiple F2 individuals, let's just check out this one individual right now and see if they have a genotype that holds pretty much to expectation. So again, let's do 1 or 2 genotypes really quickly as a demonstration. So if we look just at chromosome 1, if you start on the left side, because there's only one individual in the population at this point in time, there are 2 copies of this chromosome 1, copy 2, so half of them are red. That's 50% red. So that matches our expectation. So that doesn't make it look yet, although this is just one individual, doesn't make it look yet like there's any transmission ratio frequency distortion or segregation distortion. If we look at the right side of that chromosome, 1 red and 1 blue, that's 50% red. And then we could plot everything in between those two. On chromosome 2, the left end is 50%, the right end is half red, 1 copy is red, 1 copy is blue, so 50% red, 50% red, and so on. So we do the same plot for all the spots in between, and we get a plot that looks like this. So why does it look like this? Well, part of the reason that there are these deviations, like an extremely low value, it looks like almost all blue, and an extremely high value, entirely red, or almost entirely red, on the second chromosome, is that there is a spot on the first chromosome that is homozygous. Both copies are blue. So that's that spot. And likewise, there's a region of chromosome 2 that's homozygous in this F2 individual, 100% red, and that corresponds to that region. And this is why it's important to look at multiple individuals, because any one individual will have lots of regions of their chromosomes that are homozygous blue or homozygous red, and that will make these plots sort of extreme looking. The only possibilities are 0%, 50%, or 100% if you look at a diploid organism that only has 2 copies of one chromosome. But if we just stop here and think about how we would interpret these data, if we just only had one individual to look at, this could look like there is segregation distortion with an n of 1, just one example individual. But it would look like this individual has a blue genotype in the middle of chromosome 1 and a red genotype in the middle of chromosome 2. So what that could suggest in an extreme example is that all of the individuals that happen to get red homozygous genotypes at that spot on chromosome 1, and all of the individuals that had blue genotypes in that region of chromosome 2 didn't exist. Maybe they died, maybe the embryos were never developed. Something happened to remove all of the individuals with that particular combination of genotypes from the population. In other words, it's individuals that would have made this plot balance out. Individuals that were red, where there's a trough there, and individuals with the blue genotype, 0% red, in the middle of chromosome 2, opposite where there's a peak. Right? So that is an indication of that's a possible set of incompatible alleles. That combination of genes or alleles was never seen in this one individual. And of course, again, this is why we have to look at multiple individuals, because one individual just doesn't provide enough data to get a good sense of whether or not this is a statistically significant bias from the expected value of 50%. So here now we have 8 individuals. I've added, as it says here, 7 more random genotypes. Right? These are random genotypes because remember, the gametes that were shown, the F1 gametes, are potential recombinant haplotypes. These are not the only possible combinations of recombination breakpoints and versions of chromosomes 1 and 2 that could be put into gametes by the F1 individuals that produce them. And so these, by extension, are definitely not the only possible genotypes of F2 individuals. It's just a random set of 8 that I came up with for this example. And now, yet again, we can do the exact same analysis. We could start at, say, the left end of chromosome 1 and make our plot. So we have, how many, let's see, we have 8 individuals. So we have 2 times 8 or 16 different chromosomes. So let's count how many of those 16 are red. At the left end of chromosome 1. 1, 2, 3, 4, 5, 6, 7, 8, and 9. So 9 out of 16, or a little bit over 50% red. Pretty close to the expectation. And if you keep going through all of the chromosomes for all these individuals, this is the plot that you'll obtain. No significant deviation from 50%. The line wanders a bit around 50%, but there's nothing extreme that warrants additional attention that would suggest that there's maybe a pair of alleles that are incompatible with each other. So this is an example of what it would look like under normal circumstances where there's no evolutionary selection on any particular genotypes or combination of genotypes. So now I'm going to switch and show you an example of what happens when there is selection on a particular combination of loci. So for the purposes of this example, let's say that it's really, really bad for your health if you happen to be a hybrid that has the blue alleles on the right end of chromosome 1 and the red alleles on the left end of chromosome 2. So blue on 1, say there's a gene there, gene X, and if you have the blue version of gene X and the red version of gene Y, say, that destroys your ability to produce cellular energy, and so at fertilization this cell will never even divide into a second cell, much less become an embryo or develop into a living adult organism that can reproduce on its own. So in this example, if that was the case, if every individual that has blue on the right end of chromosome 1 and red on the left end of chromosome 2 dies before we have a chance to genotype them and measure their genotypes, then all 3 of these individuals from that F2 generation we won't see. We won't be able to collect DNA from them in the laboratory, if, for example, if they're embryonic lethal. We'll never get an adult organism to collect DNA from to measure. And what happens here is this has an effect on the graph we're going to produce, because now we're not doing the graph shown in the middle, where we were looking at the genotypes of 8 individuals. Now we've got 5 individuals that are representative of the population, but there's going to be some bias that we can see in the plot because we've gotten rid of all of the individuals that are homozygous blue on the left end of chromosome 1 and also homozygous red on the left end of chromosome 2. So let's check out what this plot looks like. So, when we experimentally determine the genotypes of all of the living organisms, 5 in this F2 generation in this case, when we genotype all of their chromosomes, we might do this, for example, by using PCR or deep sequencing technology to figure out the genotypes of each chromosome, each pair of each chromosome in each individual. We see that there's an underrepresentation of red at the left side of both chromosome 1 and chromosome 2. And if we look at the genotypes, we can see that's true. Individual 1 up here at the top is heterozygous, they're red and blue, so there's a little bit of red at the left ends of chromosomes, but most of the other remaining individuals are homozygous blue on the left end of chromosome 1. Here's another heterozygote. So there's a little bit of red left in the population, but because individuals that were, and because of recombination, which is the tricky thing in this case, individuals that were blue on the right side of the chromosomes were eliminated. That allele combination is most often associated with being red on the other end of the chromosome. See, blue, recombination breakpoint, red, blue, recombination breakpoint, red. So this form of selection, which is natural selection, individuals that have that particular combination of genotypes on 2 different chromosomes, chromosome 1 and 2, then exhibit a bias in the transmission of alleles, the transmission of red and blue from the F1 generation into the F2 generation, or at least the F2 individuals that are alive and that we can study. So this is the essence of using genotyping of hybrids to try to identify regions of chromosomes that contain genes that might be involved in genetic incompatibilities. Genetic interactions between red and blue, for example, that would kill particular genotypes of organisms or make them sick. So finally, if we turn our attention back to the allele frequency plot in the lower right, there are a couple of different interpretations of what these data mean. There is bias at 4 different loci, and we would have to do statistics to understand if this was statistically significant bias. In this case, I'm assuming that's the case. And so there are 4 loci, the left and the right ends of both of the chromosomes look like they're significantly skewed or deviating. This is the segregation distortion or marker transmission ratio distortion, depending on what you want to call it. And this is compatible with 2 different interpretations based on the plots that we see. And we've already talked about this briefly, but here's the idea. I've told you that my scenario was that blue on the right end of chromosome 1 and red on the left end of chromosome 2 was the incompatible combination. And that's that pair of alleles. So what you would expect to see if transmission ratio distortion is causing an incompatibility, and this is by no means proof of that, more experiments would need to be performed to prove that it's marker transmission ratio distortion that's causing a genetic incompatibility. But one trend is shown here. That's the homozygous blue and red is bad. So we see only red genotypes, high peak there on chromosome 2, and only blue genotypes, or mostly blue genotypes, on the left end of chromosome 2. And at the same time, we see that there's another high-low pattern where there's a peak on one chromosome and a valley or a trough deviation from the expected frequency of alleles on chromosome 1. And that is because of the individuals that were eliminated. Not only were they, as already drawn, blue on the right end of chromosome 1 and red on the left end of chromosome 2, they also happen to be, as you can see, these eliminations were also of individuals that were red at the left end of chromosome 1 and also blue on the right end of chromosome 2. And that is why that valley and that peak are present in this plot as well. So if we had actually observed these data and we saw a plot like this, we would have a lot more work to do to try to interpret which, if any, of these combinations of genes is epistatically interacting. That is, causing some sort of hybrid dysfunction that might be killing particular genotypes of hybrids. So this has been an introduction to the concepts and basic practices of detecting genomic regions that can potentially play roles in hybrid incompatibilities or negative epistatic interactions by producing allele frequency plots looking for transmission ratio distortion and then segregation distortion. Thanks for watching.